so this is our presentation on generating false memories.
Lau and her team studied how neuronal co-allocation can be used to create entirely false memories with purely behavioral manipulations. False memories are just that, false. They did not occur. Cued fear conditioning was used in the study, and this is when a neutral stimulus, like an auditory tone, is paired with an aversive stimulus, like a foot shock. An engram is a physical basis for memory. Neuronal co-allocation, the focus for this study, is the phenomenon where a neuron recently recruited to an initial engram is left in a state of heightened excitability, making it more likely to be recruited to a second engram, leaving the events linked. Previous research suggested that normally advantageous functional memory processes can sometimes go awry, leading to things like false memories. Research by Lau and her team in previous years discussed co and disallocation and how these processes directly affect memory formation and recall. And in this study, they aimed to fill the gaps in the literature by using behavioral manipulations, as opposed to optogenetic and non-behavioral methods, to describe how false memories are created, with an emphasis on the neurobiological mechanisms behind them.
So the methods and materials used in the study consist of adult mice that were both male and female, and F1 hybrid just means that the mice were offsprings of two different inbred stains since wild-type mice were used. Engram neural manipulation strategies was the method used of this study, so it was just manipulation through chemical agents that were used to manipulate and measure the neural activity amongst the mice. Manipulation forms consisted of optogenetic tools, light-sensitive proteins, and the viral vectors, which were known to help with gene expression and gene delivery amongst the optogenetic constructs. So here are the constructs listed below, the vector that was used. And priming was done before for the auditory stimulus to see if the influence it had on the allocation to neuron engrams. The mice also went through a surgical component where they were given atropine sulfate, chloral hydrate, or isoflurane and meloxicam, which was used for anesthetics and pain management during the injecting process and targeting process of the surgery. The mice were also placed in stereotax, which helped target areas of the brain region for the injecting process. Behavioral procedures focused on fear conditioning, which were used for mice that were placed in the conditioning chamber. They're presented with auditory tones of CS1 and CS2 that's listed in the study. Foot shock was associated with that and fluorescent lighting, and the freezing measured the neural activity of the memory after the conditioning process occurred.
In this study, mice exposed to a neutral tone CS2 3 hours after fear conditioning showed more freezing than those exposed 24 hours later. When exposed to CS2 24 hours later, the mice did not show the same fear response and treated it like it was a new sound. This highlighted how timing influences the interpretation of new fear events. Optogenetically activating engram neurons before training influenced the formation of false memories by associating a neutral stimulus with the fear memory. Increasing the excitability of these engram neurons led to false memory formation. However, when the neurons were inhibited during the CS2 exposure, the false memory did not form, and reactivating these neurons had no effect. Usually, no false memory forms when CS2 is presented 24 hours post-training, but artificially activating engram neurons after 24 hours can still create a false memory, which shows the importance of neuron excitability in memory formation. Lastly, the study revealed that both real and false memories rely on the same neurons, and inhibiting these neurons prevent both. This supports the idea that false memories form when a neutral stimulus is linked to a fear memory through neuron co-allocation.
So the first conclusion that we have is that memory distortions may be a result of normal adaptive memory processes. This means that false eyewitness testimony or inaccurate or implanted childhood memories or just everyday lapses in our ability to recall events of the past can just be due to a bug of the memory. Second, we have temporal proximity of similar events can result in the events becoming linked. So they can be connected by virtue of a co-allocation of neurons to overlapping engrams. This means that the allocated engram neurons remain more excitable after the first event, and if a second related event happens during the time of excitability, the engram neurons for the first event are allocated as the engram supporting neurons for the second event, and this causes the events to become linked. Third, we have the creation of false memories is due to the excitability-dependent co-allocation mechanisms mentioned above. So linking up this mechanism is dependent on neuronal excitability. So decreasing excitability of neurons for the first event will prevent false memory formation, whereas artificially increasing excitability will facilitate false memory formation. And last, we have artificially creating or preventing formation of false memory is done by mimicking endogenous excitability states of engram neurons.
Two limitations are temporal constraints and animal model limitations. So is the conclusion supported by the paper? Strong experimental evidence shows that false memory can be induced through neural excitability-based co-allocations. Findings align with memory linking and engram researches in neuroscience. So yes, the conclusion is supported. How does our article relate to what we have learned? Starting off with the brain regions, the hippocampus and amygdala, critical for memory consolidation and fear learning, as well as the temporal lobe, as it supports sensory integration and episodic memory. For synaptic plasticity and memory co-allocation, the Hebbian learning explains false memory formation. For neurobiological approaches, used optogenetics directly manipulate neural excitability. Synaptic changes measure how false memories form